Question 13 is showing the stage micrometer and also the eyepiece reticule in the figure. So this type of questions is the type of question that use the eyepiece reticule to measure the cell. So it is measuring cell using the eyepiece reticule. So to solve this question, we will follow the 3 steps. For this question is a bit special because instead of asking for the size of a cell, this question is giving us the size of the cell initially, and then they are asking the number of the pitch on the eyepiece reticule that can cover these 50 micrometer. So this is the only thing that is special. Well, the rest of it, it is still following the 3 steps. So step number 1, you still find the 1 pitch on the stage micrometer. So to find that, we have to look at the info given by the questions. So this is a stage micrometer with the scale of 1 cm scale. Yeah, the stage micrometer is having a 1 cm scale. And this is not a special stage micrometer, it's just a normal stage micrometer. So that's why it will be 100 pitch on the stage micrometer equals to 1 cm. So we have to convert the cm. So cm convert to mm, we times 10. So it will be 10 mm. Then from mm, we convert to micrometer. We will times 1000. So it becomes 10,000 micrometer. So this would be 1 cm convert to micron. So with that, 100 pitch on the stage micrometer equals to 10,000 micrometer. So 1 pitch on the stage micrometer will be equals to 10,000 micrometer divided by 100. So 1 pitch on the stage micrometer is equals to 100 micrometer. So that is the end of the step number 1. Yeah. So now we know that from here to here, this is 1 pitch on the stage micrometer is equals to 100 micrometer. This is what we have calculated out. So maybe I will just draw it out. So this will be equals to 100 micrometer. This is 1 pitch on the stage micrometer. Then next, we'll move on to the step number 2. So step number 2 is we have to find 1 pitch on the eyepiece reticule. Yeah, 1 pitch on the eyepiece reticule. So meaning that we want to find this 1 pitch on the eyepiece reticule is equals to how many micrometer? So to find that out, we have to find the 2 overlapping line between the stage micrometer and the eyepiece reticule. So this is the line, in the figure, there's no other choice. Yeah, there's only 1 line, 2 line of the stage micrometer. There are only 2 lines here, so we have to use these 2 lines. So we have to see which line on the eyepiece reticule that it overlap with. So for some of the figure, you will see that the stage micrometer line is so thick. Yeah, and the line for the eyepiece reticule is so thin. So which line we are going to use when we want to see the overlapping line? For example, over here, yeah, you can see that this is 1 line of the stage micrometer, and it covers 3 lines of the eyepiece reticule. So which one we should use? Same thing goes to this one. This is the 1 line of the stage micrometer, and the 1 line covers 3 lines of the eyepiece reticule. So which line we should use? So to be safe, normally we will use the one that's at the center. For example, you can see that 1, 2, 3, 3 lines of the eyepiece reticule. We will choose the one in the center. So same thing goes to here. There are 3 lines. We will choose the one in the center. Okay. So with that, that means that this is 1 line of eyepiece reticule, second line, and third line. So we will choose the second line. So that means that it is from 32. Yeah, it is for the eyepiece reticule is 30, 31, 32. So we will state that this line 32 is overlapping with the stage micrometer line. While this one, 71, 72, 73. Yeah, 70, 71, 72, 73. So for this one, we will state that 72. Yeah, line 72 of the eyepiece reticule is overlapping with the stage micrometer line. So from 72 to 32 total, there are actually 40 pitch on the eyepiece reticule. Yeah, there are 40 pitch on the eyepiece reticule. Yeah, from line 32 to line 72. So it is total 40 pitch on the eyepiece reticule. So based on this info, there are 40 pitch of the eyepiece reticule equals to from here to here. Yeah, it equal to only one pitch of the stage micrometer. Yeah, only one pitch on the stage micrometer. So it's equals to one pitch on the stage micrometer. So we have calculated just now, one pitch on the stage micrometer is equals to 100 micrometer. Yeah, so that's why we have to times it out. So 40 pitch on the eyepiece reticule equals to 1 times 100 micrometer. So one pitch on the eyepiece reticule equals to 100 micrometer divided by 40. So that means that one pitch on the eyepiece reticule is equals to 2.5. Yeah, is equals to 2.5. So that is step number 2. So with that, now we know that one pitch on the eyepiece reticule is equals to 2.5 micrometer. So we know the size here. Then step number 3. Step number 3 is now we can use the eyepiece reticule to do the measurement. That is step number 3. So step number 3, we will measure. Yeah, we will measure. So to measure, well, normally the size of the cell, yeah, the size of the cell normally is equals to, we will see how many pitch it will cover. Yeah, number of pitch on the eyepiece reticule that will cover the cell times the length of one pitch on the eyepiece reticule. Okay, but as I mentioned at the beginning, this question is a bit special. Instead of asking for the size of the cell, they are giving us the size of the cell, but they are asking for how many pitch on the eyepiece reticule that will cover this cell. Okay, but since we have this equation already, then yeah, we will just substitute whatever we have, yeah, whatever info that we have. So based on the question, the size of cell is 50 micrometer. So size of cell is 50 micrometer. So number of pitch is actually what we want to find. Yeah, number of the pitch on the eyepiece reticule is what we want to find. Okay, so we will just write it down. Number of the pitch on the eyepiece reticule. So times the length of one pitch on the eyepiece reticule. This one is what we have calculated just now. 1 PE is equal to 2.5 micrometer. So it is 2.5 micrometer. So with that, we rearrange it. So that means that 50 micrometer divided by 2.5 micrometer equals to number of pitch of the eyepiece reticule. Okay, so with that, number of pitch of the eyepiece reticule that will cover this one cheek cell, that will cover this one cheek cell equals to 20. Okay, because 50 divided by 2.5, yeah, so it is 20